Celecoxib is a selective nonsteroidal anti-inflammatory drug that inhibits COX-2, which is responsible for prostaglandin synthesis. Unlike other NSAIDs, it minimizes gastrointestinal side effects by targeting COX-2 instead of COX-1. While celecoxib is widely prescribed for pain and inflammation, its precise protein targets in the central nervous system have remained unclear. Target deconvolution is the identification of specific biological targets that a drug interacts with in the body. It aims to uncover the molecular targets responsible for the drug's effects. We developed thermal proteome profiling to function as a label-free method for identifying drug targets based on changes in the thermal stability of proteins in response to drug binding. Proteins bound to drugs display increased stability, making them more resistant to heat-induced unfolding. In our study, we used TPP to investigate celecoxib's targets in the rat hippocampus. We identified 44 off-target proteins associated with signaling pathways linked to neurodegenerative diseases but also cancer. Our findings, backed by computational evidence, suggest potential repurposing opportunities for this NSAID in other areas. To verify the targets, we compared them with established celecoxib targets in multiple drug target databases, indicating celecoxib's potential for multiple brain targets. Additionally, we investigated the structural similarity between TPP-identified proteins and reported targets. The results indicate promising overlaps, further confirming the identified celecoxib targets. The enrichment analysis of TPP-identified proteins showed associations with various diseases and pathways, including muscular stiffness, neurodegenerative diseases like Alzheimer's, epilepsy, and breast cancer, suggesting potential therapeutic implications of celecoxib in these areas. Next, we examined gene ontology annotations and identified significant processes affected by celecoxib, including SRP-dependent protein targeting and Rab protein signal transduction, and annotations related to mRNA catabolism, protein modification, and immunity, suggesting that celecoxib's effects involve post-translational protein modifications in G-protein-related signaling pathways. We also investigated PTMs in the TPP-identified proteins and found enrichment of acetylation, prenylation, and phosphorylation, emphasizing their role in the thermostability of proteins. Notably, citrullination emerged as a specific PTM enriched in celecoxib targets. This suggests that PTMs may play a role in regulating the activity of these proteins in response to celecoxib treatment. Our research uncovers the potential of celecoxib, shedding light on its specific protein targets in the CNS. By using TPP and computational analysis, we provide new insights into the broader impact of celecoxib on cancer and inflammation pathways, in addition to neurodegenerative diseases. These findings pave the way for future drug repurposing strategies, opening up new avenues for improved therapeutic approaches. Thank you for watching. If you want to read our paper for yourself, we've provided the link for it in the description below.